Welcome to the Unit 3 Human Digestion Lab. This walkthrough is going to focus on Day 2, which is Part E, where we record the results of our experiments 48 hours after. We're going to start by looking at the milk digestion tables, but before we do that, we need to enter in the code we got from yesterday and hit. We're going to be focusing on the milk digestion tubes, these first 4 we set up. Remembering that when milk is digested, the lactose sugar is split into glucose and galactose. We're going to be using a glucose strip tester to determine if glucose is present. So now we're going to use these strips here, and we're going to compare them to this chart. I'm going to do one simulated with you. Just a reminder, you have to use the tweezers to pick up the test strips. We're going to use these test strips this time. They start blue. You just stick them in, click the mouse button, and then you can hang them back up here. So they're either blue to start with, or they're going to change a color. As you can see, I've already tested all 4 strips for us, and as I said, they begin blue, and they'll turn to the green-orange side, depending on if and how much glucose is present. So you can see that 2 of our test tubes, the first 2, don't show any indication of glucose, and the 2 on the end show quite a lot of glucose present. Refresh your memory and look at the 4 test tubes here. Remember, the first 2 did not show glucose present. The last 2 did. We did an entire lab on this for the lac-operon procedure. Remember, the enzyme that breaks lactose down is called lactase, and it breaks it down into glucose and galactose. So let's write in our test strips. No glucose for the first 2. No glucose for the second. When we fill in this last column here, we just have to decide, was the milk properly digested down into glucose? So as long as there was lactose present, you'll be able to see glucose in the solution. So no for the first 2. Now the second set of 2, so the third test tube does, and the 4th test tube does. The difference between the 2 is the presence of acid. Now remember, bacteria can break down lactose with lactase even without stomach acid, because bacteria, our lac-operon lab was based on, they don't have stomach acid. So even without the presence of HCl, as long as lactose is present, we'll have digestion happen. And that's the end of this portion. We're going to move on in the next video to E protein digestion.